Thank you so much for the invitation. I'm really happy to be here again. So I'll just continue my lecture too, I guess. So my name is Sharmila Venugopal. I am from the Integrative Biology and Physiology department here at UCLA. My research is in neuroscience, and I have started the Neural Dynamics group, where we combine experimental and computational approaches, and we study neurodegenerative disease dynamics. Just to give some of you who may not have been here last week, just to get started, like I said, we basically look at diseases, and I don't need to say this to this group, computational methodologies and dynamic modeling are essential tools when we study these complex diseases and their mechanisms that are occurring at multiple spatio-temporal scales. So this is a slide that I added to kind of summarize what we are looking at. So we are broadly looking at a specific neurodegenerative disease, but we are specifically focusing on how the neurons in the brain that are dying due to the disease chronically, they're different from the surviving neurons. So we think that by understanding the functional signatures, circuit signatures, molecular signatures of these two very different kinds of neurons, we may be able to understand the disease better and identify windows of opportunity for intervention. And to do this, it is inevitable to use dynamical approaches as well as other types of computational approaches. And the goal for studying the dynamics is to discover the signatures, the dynamic signatures pertaining to the stability properties of these cells, circuits, and so on, as well as their response to uncertainties. And disease as such itself could present uncertain situations for these cells, and that studying those responses would help us understand the disease better. And a part of the goal is also to understand the rules by which the homeostatic control is made. And this is a very difficult question to answer, and we are trying to kind of come up with, our theories and ways to kind of summarize what kind of homeostatic control is present in the system. So I'm happy to discuss any of these things in detail, but that's kind of the gist of where we are with this. We do use a combination of approaches. Like I said, we do experimental work, computational modeling, simulations, and theory. And also today, I want to kind of share with you this use of this closed loop interfaces where the models come in closed loop with real experiments, and this happens in real time. Last time, I already presented a list of complementary computational approaches that we use with our experimental work. Mainly, we have used the dynamic modeling, and it's all data-driven modeling approaches. And the uniqueness is really has to do with the multiple scales that we target. We model them at cellular scales, network scales, and also we are keen on looking at coupled subsystems, for example, integrating neurons with muscle cells, or integrating, neuroinflammatory versus neural excitability systems and things like that. And we are very excited about the real-time interface that I mentioned. And we also time and again invent, our ways of using quantitative approaches coming from information theory, image processing, etc., to study these cells and network properties depending on what kind of data we have. So that's a summary of that. And these are things that I went through last time. So I'm going to quickly go through some slides. I just want to point out one area here. The big idea that I presented last week is how we have examined at an early stage of the disease in a mouse model, what are the different physiological signatures of the cells that are vulnerable to the disease. And in doing so, we have discovered that there are cells that are other than the ones that are dying that are also changing in the disease very early. And one such type of cell is these sensory cells. And we are studying a motor neuron disease in which motor neurons die, and sensory and motor are coupled together, and they drive these very important reflexes that we all use to kind of, do any kind of movement for that matter. So our big finding here was that the sensory neurons have these very irregular patterns of action potentials that the neurons show. We call them spikes. And this red here is the one in the diseased mouse, whereas the blue trace here shows the regular mouse or its wild type littermate. As you can appreciate, this is very rhythmic. This bunch of blue here is essentially individual spikes clustered together. So there is a long delay here in between such bursts of activity. We call this rhythmic bursting. And I'm highlighting this because we did a lot of work trying to understand what are the molecular basis of why this transforms to something like this. And we then used mathematical modeling and also real-time, closed-loop approaches to fix this kind of aberrant behavior of these cells to making them look more like this. So so that's kind of one of the key ones I want you to remember as I present the other things. This is all, once again, something I spoke last week. So I'm going to kind of quickly go through this and tell the interim summary as so far. So like I said before, we are studying a neurodegenerative disease, which is a motor neuron disease. So previously, the main notion for this disease was that these specific types of neurons that connect the nervous system to the target organs that are the muscles and are responsible for integrating all of the brain inputs and convey that information, convert that information to movement. It is the job of these motor neurons. So in this disease, these neurons die, and that's kind of the prevailing theory that existed. And now with our work and many others, we have a much broader understanding of this disease. And now it is, we have provided evidence that it is actually a circuit specific disease. So from a biological standpoint, this from our series of studies that we have done, it's a significant contribution, and there is a lot to be done going from there. And today what I want to do is focus a little bit more on the dynamics aspect of what we have found and also alluding to the neural information aspects as well. And there are many questions that have risen from these studies and many other similar studies. These have to do with understanding the protein networks, the cellular interactions, and also, once again, discovering the homeostatic rules of how disease modulates the nervous system in a longitudinal manner. So these are all question marks because these require a lot more experiments as well as computational work to be combined to discover more in that area. So, so to start off, this is a busy slide, but I really, it's one of my favorite slides because this is the work that I did with a couple of years ago or so, where we had some really beautiful insights come out of the computational modeling that we did. So what this entails is that basically we looked at the sensory neuron that was changing in the disease. And we found that a specific type of ion channel that is important for the generation of these spikes or action potentials in these neurons was downregulated. So we were curious to see what kind of roles does this ion channel play in this neuron's ability to encode information and to generate these rhythmic spikes. So what I show here, this purple part here shows you the model that we generated for the type of currents that are missing in this neuron, not completely missing, but are downregulated or reduced. So here I show the rhythmic bursting in this model cell, and there is a specific type of component of this channel current, which is called a resurgent current. That was very, we got very curious about it. It was reduced in the diseased mouse, and we were curious to see what role did that play. The disturbance in the rhythmic pattern of firing that we saw in this mouse, was it corrected by this current, or did it have anything to do with that? So that was our question that we were trying to answer. So we generated this mathematical model of the neuron that reproduced this beautiful bursting pattern. And then we actually, to make this neuron go crazy, meaning, generate these irregular spikes, we basically added some noise. So we made it look like the diseased mouse pattern of firing action potentials. And then we added the model current that we had built. So notice that how the inclusion of this resurgent sodium current actually fixed the irregular pattern that we saw here. And then, of course, this is a beautiful explanation of how this happens. I won't take you through the whole details of this. I don't want to bore you with all that details, but I will say that, we use this bifurcation analysis from dynamical systems. And the big idea here is that this green region that you see, which we call the region of attraction, and that region of attraction around this stable, steady state is rather small. And before, every time this neuron tries to generate these bursts of action potentials, it has to pass through this regime and cross this threshold, and then it starts to, do its spiking activity. Now, with the addition of this resurgent current, we notice that this green region grew really big, meaning that there is a region of noise tolerance. In other words, if there was any noise here, at the beginning, in between these bursts of activity, the neuron became rather resistant to those noisy inputs, and therefore would not generate these abrupt spikes. So this was a really interesting finding for us. And we actually set out to test this experimentally as well. But to take home, this was a very insightful finding, such that we had a single channel that was mediating a certain pattern of activity in these neurons, and different components of the currents mediated by this channel was causing different types of properties in these sensory neurons. For example, one of the types of components, the persistent component, helped the neuron in its input selection. Since these are sensory neurons, they have to be selective to the type of frequencies of the inputs that they have to choose from. So this basically helped the neuron tune in to the right kind of frequencies. And then this other component helped eliminate some of these, noisiness in that encoded bursts of activity. So overall, we thought that this type of current was helping the neuron to generate rhythmic bursting, which it is expected to do. And in the disease, when these currents are reduced, it is possible that these neurons lose the ability to properly encode information. So that was really a key insight from there. And now I will move on to kind of giving you a flavor for how we use this closed loop approach to put the model in the loop with experimental ongoing experiments. So this is, once again, the top part here is to illustrate the dynamic model. And when we put the resurgent current, the blue trace here shows you that the current turned on, and you can see correspondingly the change in the pattern of firing here. And what was really cool, and this we did in collaboration with Dr. Ricardo Olchisi's lab in the medical school, and his lab member, Dr. Antonios Pantasis. He's a faculty member now elsewhere, but he was really a key person to help us get this going. And Soju Seiki here was my postdoc who conducted these experiments. So essentially, the way this worked was we make the computational model sit here in the computer. This is the channel model, and the experiment gives us the readout of the voltage from this neuron, and we manipulate the neuron by injecting a certain amount of current, and this current could be coming from this in silico channel. It can also have step-like inputs and things like that. So the idea here is that as we are recording the voltage in this neuron, that voltage is fed back into this model, which then updates the channel current as though it is a knock-in channel into the real neuron. And then that current then, once again modifies the neuron's properties, and then we see, the type of response that we saw in the actual model neuron. But this is a real neuron with a manipulation coming from the model channel. So this was really cool that we were able to mimic what we saw in simulations in actual experiments. So we then used this to test to see if we can modify the amount of information present in the spikes. So for instance, here, these black clusters here are actually many rasters, this corresponding to the neural spikes. So these are all spike rasters. And when we put the noise, you can appreciate that there is a lot of these arrhythmic kind of pattern here. And then we add the type of current that we are testing, and we increase the intensity of that current, and we see that the irregularity starts to disappear. And we reproduce the same thing with this closed loop experiment. So this is a real neuron with its rasters. And then we actually added some noise as well to make the neuron even more noisy. And then we added these currents to fix that noisy pattern and make it a much more rhythmic pattern. And this is just showing you the intervals between the spikes, another way of looking at the same data here. And we measured things like entropy and this, variance of these spike intervals and things like that. And then we saw that we could indeed manipulate the information present in the spikes and actually fix that with a specific type of ion channel current. So this was also very exciting for us. And finally, we put that in practice to see if we can correct what happens in the disease. So we took a neuron which had irregular bursts of activity in a real disease mouse, and then we incorporated these model currents, and we tested different components of these currents, and we saw that only one of these components, the resurgent component, was capable of fixing this rather very irregular raster here that you see here to showing more rhythmic unified values here as you increase the intensity of this current. And then when you removed it, the irregularity came back, verifying that it was indeed a response that we saw due to our manipulation. So this, from a physiological standpoint, this was a insightful finding and a demonstration that a single channel current can indeed produce very different kinds of effects on neural information, the burst patterns, and the stability properties of these neurons. So together, those studies led to the making of this later network model. So here we tested to see if the disturbances that we saw in the sensory neuron, as well as the disturbances that we had seen in the motor neurons previously that I described last week, we put that together and made this into a what we call a monosynaptic reflex network, meaning a one-stop connection here. And this type of network is very important for stretch reflexes. And in the disease, when we see these abnormal patterns, and correspondingly how the motor response changes is what we tested here. And notice that if the interval between these bursts in the sensory was quite large, the motor neuron kind of accommodates its spikes and it shuts off, more or less being synchronous with the sensory. But when these intervals rather become shorter and irregular, the motor neuron it becomes confused. In other words, some of its own properties present in the dendrites of these motor neurons, they fail to accommodate, and they continue to make the motor neuron keep firing. And this type of firing of the motor neuron can activate the muscles continuously, even though it may not be required to do so, and lead to dysfunctional type of muscle activation, which we call fused self-sustained activity. In this case, these can basically result in hyperreflexic or muscle twitching that is observed in these patients early on during the disease development. So overall, that's kind of my story for now. I'm not sure if I presented all the data related stuff that we did, but I do want to thank all my collaborators who have been extremely supportive, without whose help and timely resources, all that it wouldn't be possible to complete this work. The postdocs, and there are several undergraduate students that are besides this list here. And special thanks to Dr. Garfinkel in our department. Like I said last time, many productive discussions have, it's been very useful, especially for the modeling components of this project. And I also am thankful to the support and the new communities that I've been engaged with. I did want to put another additional slide here to kind of highlight where we are going with this. And in terms of modeling, of course, there's a lot to be done. We are looking at some of the data science approaches, trying to kind of mine some of the databases that are existing. We are continuing to do dynamic modeling. And in regards to that, we are looking at optimization techniques to see whether, the disease does a different kind of optimization compared to the normal, and what kind of fits would come out of there. We are trying to generate protein interaction networks. So some of the network theories might come of use there. And of course, with our goal of trying to understand the rules of homeostatic regulation, control systems theories would also be applicable. And also I want to note that we have a neuroinformatics project that we kind of began. It's very much in the preliminary stage, so I didn't want to bring any specific data from there. But the goal there is to now address the big data problem in neuroscience. As all of you can appreciate, now neuroscience is not neuroscience only. It has now become neuroglial science. In other words, there are all of these other types of cells for whom there are ways to measure physiological data, ways to measure structural data, morphological data, and these are all high throughput data. And we're trying to sort of make connections with other groups as well as with organizations that help develop standards and best practices for basically assembling these data sets, analyzing these data sets, documenting them, archiving them, and so on. And a lot of this data comes in the form of image data. So these are all image time series. So there's a lot of information in every single frame of this image. So in the past, I was involved in developing a software that enabled some of this image analysis for calcium imaging in astrocytes. So I want to kind of continue doing that and also include many of the newer things that have come in this area. Is improvisation in this area. And the goal really, hopefully in the future, we will be able to come up with a computational ontology of the multicellularity of the brain as we know it. If anybody is interested, students especially, feel free to contact me and we can talk about how we can engage you with our work. Any questions, please feel free to ask.
Thanks, Sharmila. That was fascinating. I think you've convinced me if I was going to do my career over again, I'd become a neuroscientist for sure.
Fantastic. Thank you so much. That's very flattering.
But if I can just, what's really amazing to me is how you can connect, because the signals are electrical, you could connect them to man-made hardware, right? The cells, and so the man-made hardware is run, interprets signals, and then provides feedback based on a mathematical model that is informed by the biochemistry of receptors and channels. I mean, we're channels,
Exactly.
right? So that model is, you know, it's just a few equations, right? It's not a very complex model, but the parameters, I imagine the parameters do vary from cell to cell, right? If you're doing that experiment on 10 different cells, presumably, so is there a way of actually tuning the parameters in a cell-specific manner, like for each experiment?
Yeah, you asked an excellent question. I really appreciate it. I mean, it's a fantastic question. So one of the things that I've been thinking about very much is, you know, each cell has its own characteristics, like you said, and we can come up with a transfer function for each cell as we measure these parameters. As we measure voltages from these cells. And one thing I would be very interested, and I'm actually kind of one of the companies that make these systems, the dynamic clamp systems, they have been approaching me time and again to kind of give them input and feedback, how to improvise their system, use their system and such. So one of the things that came to my mind is you could use an optimization approach to tune the parameters of the model to suit a certain type of transfer function. And then you can use that set of parameters for your ion channel. So one other thing I want to mention is that we don't have to be limited to a single ion channel like we have it. We could have an entire neuron. In fact, my goal, my dream experiment was to have the sensory neuron as the dynamic clamp model, and then the motor neuron being, you know, experimentally evaluated, and then you provide the full sensory input to the motor neuron. So that, you know, it's not just one neuron, you can also have a network of neurons in the computer. So this is a very powerful technique if one can use it very creatively.
Mm-hmm.
And can be, especially with our goal of studying dynamics and information. And this would be excellent to clarify how physiological and biological properties and parameters translate into dynamics.
Mm-hmm.
So I think that was very, yeah.
That's great. Mm-hmm. Any other questions?